Welcome, Junior Tuckies. My name is Mrs. Ramlal. This is a very special presentation in which I have prepared for the grade 12s as they are a few days away from their final exam. This is work matrix that you need for paper 1. So let's look at your topics. Now, for your topics for paper 1, you must know reproduction in invertebrates, human reproduction, responding to the environment in humans, responding to the environment in plants, endocrine and homeostasis. These are your marks, 8, 41, 54, 13, 34, and your paper will be out of 150. Right. The questions you're going to be asked is you will have section A, and section A is your terminology questions, your matching column statements, your short answer questions such as multiple choice. So all of that will be 50 marks. Section B will be 2 questions of 50 marks each. And here you'll have a lot of sub questions and it will be out of 100. Right. Grade 12s, when you're preparing for this paper for Friday, make sure you work with your exam guidelines. That's very, very important. Everything in the exam guidelines, make sure you know it. Right, let's get down to some skills that I think you must know before you write your exam. Right, know your skills well. Make sure you have a heading for any task that you're given in the exam. Right. My best advice, make sure you know how to calculate percentage increase and decrease. If you don't know how to calculate this, you can always go to the Junior Tuckies lessons, the grade 10 skills lessons, and you can watch it there. Read the questions carefully. Do not get confused between percentage difference and percentage increase or decrease. Some of you, when you see percentage difference, you immediately want to do the percentage increase and decrease. For percentage increase and decrease, it is the same formula. Difference over the first number multiplied by 100. Don't get scared if the first number is a larger number. Do not get scared if that you calculate your percentage is in hundreds. If you have hundreds of percent, 400%, do not get scared. That is possible. Right, let's look at graphs. This was a graph that was asked in the Gauteng prelim last year's paper, 2021. Now, the learners had to draw a graph of the measurement of the fetal head circumference from 14 to 40 weeks. Most of them wrote over time. That's incorrect. So in the heading, you must have 14 to 40 weeks. That's what is required. Okay. Now, the graph from this year's Gauteng prelim paper. The learners had to draw a graph to show the average heat release from the human skin where temperature ranged from 16 degrees and 44 degrees. Now, you can't say over different temperatures. It's specifically from 16 to 44 degrees. So for me, my best advice I would give you is make sure, don't write over time. Don't write of different temperatures. Be specific in this case. Let's look at scientific method. You must know how to identify the dependent and independent variables. Fixed variables, if you are asked, must begin with the word same. Be specific when you're writing the fixed variables. Don't just say same person. Say same person measuring the diameter of the pupil. Relate this to the experiment. Say same instrument to measure the diameter of the pupil. And something that I've realized this year, do not say same amount of water anymore. The examiners are looking for same volume of water. Try not to use environmental conditions. It's too vague. Please, my advice to you is read the experiment. Write the variables that can be applied to the experiment. If there's no measuring needed, then don't write this as a fixed variable. When it comes to calculations, always show your calculations. Always include units in your answers. My advice is have the raw answer and then round off the answer. For example, here 40.56%, and then you round it off. Okay, you don't know what answer is required in the memo. In this case, you will be awarded the marks. If you are asked a pie chart, always include calculations. Calculations usually count for 2 marks. Right, reliability. You may be asked, how can a person ensure reliability? In this case, you can say increase the sample size, repeat the investigation, or take random samples. But when it comes to increase the sample size, you have to say increase the sample size of the number of plants, or increase the number of plants, or increase the sample size by using more plants. So you need to be specific and make sure you relate it to the experiment that you're given. You might also be asked, how has the person ensured reliability? This means reliability is already ensured in the text. So from the text, you have to say how the person ensured reliability. So in this case, the investigator used 100 piglets. That's a large sample size. Or performed the experiment over 30 days. Make sure when you are reading the question, read carefully. Validity. Now, validity, you always say, yes, is to make the experiment fair. But how do you make an experiment fair? You make sure you have fixed variables. And remember, the fixed variables always start off with the word same. And these fixed variables must stay the same throughout the experiment. Make sure that you read the question carefully. Now, they might not ask you just fixed variables. They might ask you a specific type of fixed variable. Now, what do I mean? I've taken this example from one of the prelims. State 2 variables that should have been considered about the zinc supplements to ensure validity of the investigation. So you see, here they're not talking about the fixed variables only of the entire experiment. They're only asking fixed variables by relating to the zinc supplements only. So here you would have said same concentration of zinc supplements. And you're not going to say same amount, remember? You have to say same volume of zinc supplements. Try all the time to replace the word amount with the word volume. I've taken this example from the Gauteng prelim paper. It was a very nice question. So let's go through it together. One of the symptoms of COVID-19 is a loss of taste. Many patients take a very long time to regain their taste after recovering from COVID-19. A group of learners decided to perform an investigation to compare how the time after infection with the virus influences the amount of taste recovered. They got a number of learners who had recovered from COVID-19 and 2 learners who had never been affected to volunteer for the investigation. They followed the procedure below. They produced salt water solutions of different concentrations as described in the table below. A description of how the salt water solutions were produced. So there's your bottle sample, A to E, salt concentration. It was 50 ml of water with no other addition, it's pure water. 50 ml water, 1 teaspoon of salt. 50 ml of water with 2 teaspoons of salt. 50 ml of water, 3 teaspoons of salt. 50 ml of water with 4 teaspoons of salt. Now, pipette droppers were used to place 3 drops of one solution on the tongue of the participants. The participants were asked if they could taste the salt. After 5 minutes, 3 drops of another solution were placed on the tongues of the participant. They were again asked if they could taste the salt. The process was repeated until all the solutions were tested on each of the participants. The solutions were given to volunteers in a random order. Now, how in this experiment, where is now the aim? So immediately it says here, perform an investigation to compare how the time after the infection with the virus influences the amount of taste recovered. So this is now your aim. What is found in your aim? Your dependent variable and your dependent variable. So independent variable and your dependent variable. Here in this case, the question, one of the questions is they're going to ask you, what is the dependent variable? What are you measuring? You are measuring the amount of taste recovered. Here it is here. Okay, so make sure when you're given an experiment, you read the top introduction. Usually where they talk about to investigate or an investigation, that's usually where the aim is. Look for the aim, because in the aim, that is where the dependent and independent variable is. If you look here, they've given you some fixed variables. They placed 3 drops of one solution. So they placed 3 drops, the same amount of solutions. They use the same time intervals. Okay, so there's your fixed variables here as well. Right, let's get down to some questions. Now let's carry on reading the results. The results of the investigation are captured in the table below. An X was placed in the box so the learners said they could taste the saltiness. So there's the participants, there's the duration since COVID. These 2 never had COVID, and there's the solutions. Right. The learners decided not to consider Given in their analysis as they thought he was deliberately, on purpose, giving incorrect answers. Hence, you can see it scratched out. They replaced him with Tomi-san, who also recovered from COVID-19 6 weeks ago. Right. So the questions they're asking is, consider the samples A to E. Which sample had the greatest concentration? That would have been sample E. That was easy to read. Greatest concentration, the one that had the most amount of salt. Identify the dependent variable in this investigation. I just told you. It was the amount of taste recovered. Identify two variables the learners kept the same. We also did that. It was the same number of droplets and the same time interval. Here you could have even said same volume of the salt solution. Not same amount, same volume of the salt solution. Make sure it's just two, your two best answers. They ask you to describe the general trend shown by the results. Okay, so as more time passes or as the duration from COVID increases, the number of samples tasted also increase. Right. Select two participants that acted as a control. The two participants that acted as a control was the two that didn't have COVID. Right. And then they ask you to give a reason for your answer. Now the reason why I'm doing these questions is because of question 7. Explain how leaving out Given's results, replacing it with Tomi-san's results, affects reliability and validity. Now, reliability has to do with number and repeating the investigation. So here they didn't repeat the investigation. Here it has to do with obviously sample size and validity. We have to look at, did it affect our fixed variables? So this is a very important question. Leaving out one result, replacing it with another. Does it affect reliability and validity? 4 marks. Right, let's go. We already said it was E. You can always go back and you can have a look at the answers. Amount of taste recovered, I showed you where to get this answer. See, the same liquid used. See, the same volume of water. The same solute used, the same number of drops, the same method of recording, the same number of participants per duration. Right, so these are some of the fixed variables. As a duration, so they asked you what was the general trend for the results. As a duration, since the infection increased, the greater number of samples can be tasted. Or you could have said the greater the time after infection, the more taste is recovered. Or the shorter the time after infection, the less taste is recovered. Any one that you would have had. Then which are the two that act as controls? Ritabila and Martinez. If you look at your results, they did not have COVID. And there's your results for number 6. Now let's look at 7. Replacing Given's results with Tomi-san's. Did it affect reliability? No. You're just taking one result and you're replacing it with another. So the sample size remained the same. The other answer they wanted for the reliability increased because the average could have increased. The average that you calculated could have increased. Did it affect validity? No. All the fixed variables in the beginning of the experiment were not affected. So remember, validity. For validity to be maintained in the experiment, the fixed variables must remain the same throughout the experiment. And by changing one result for another, it did not affect the control variables at all. So make sure you learn how to answer questions like number 7. It's a very good question for you to get. Right. Let's look at the following. You must know the following very well and don't get confused. Right. Let's look at corpus luteum. Remember, the corpus luteum is in the menstrual cycle. Okay? Corpus callosum is in the brain. Know the functions well. Chorion is a membrane in the amniotic egg or in placental development. Choroid is a layer found in the eye. Don't get confused. Umbilical artery carries deoxygenated blood away from the fetus. The umbilical vein carries oxygenated blood to the fetus. Cerebrum, cerebellum, know well, don't get confused between the two. And parasympathetic and sympathetic, also very, very important. Now are starting to show up more in past papers. Oval window and round window. We'll cover it later, but don't get confused. And then glucose. You must know the difference between glucose, glucagon, and insulin. Right. Let's look at some content now. Right, let's look at reproductive strategies. It's 8 marks. Ovipary, an organism that lays eggs, say, outside the female's body. Vivipary, an organism that gives birth to live young. Ovovivipary, learn how to spell this properly. An organism that forms the egg inside the female's body, the egg hatches inside the female's body, and then she gives birth to live young. Altricial development, where the organism or the bird that forms is less advanced, has closed eyes, is dependent on the parents, and if it's a bird, has no feathers. Precocial development, the organism or the bird that forms is an advanced state, has opened eyes, is independent, and has feathers. Know how to label the amniotic egg, as well as you must know the functions of the structures as well. Right. Let's look at reproduction in humans, 41 marks. From your exam guidelines, know spermatogenesis and oogenesis. Spermatogenesis, you know only meiosis occurs. Oogenesis, mitosis, and meiosis occurs. Know how to draw an ovum, a sperm, and know how to draw or write the adaptations of the ovum and the sperm. Now, an adaptation is how the structure helps the function. So if you don't know an adaptation, just link the structure to the function. Learn the male reproductive system well. Know the epididymis. Many cannot spell this. Also know that sperm can only be produced at 2 degrees below body temperature, and that's why the testes is outside the male's body. Learn the hormones, follicle stimulating hormone and luteinizing hormone. Estrogen, you should know, makes the endometrium lining thick, vascular, glandular for implantation. Progesterone will make the endometrium lining more thicker, more vascular, more glandular for implantation. Make sure you know the negative feedback mechanism of progesterone and FSH. High amounts of progesterone inhibit FSH. No follicle stimulating hormone is produced. No ovulation, no pregnancy. Then, the parts that make up the placenta, you should know, is the chorionic villi, that's the fetal blood vessels, and then the endometrium, the maternal blood vessels. Remember, there's always a sinus between these 2 blood vessels. These blood vessels never actually touch. Then, when it comes to embryonic development, they may ask you from fertilization till implantation. Till implantation, this is the answer. Diploid zygote undergoes mitosis to form a ball of cells called the morula. The morula undergoes more mitosis to form a hollow ball of cells called the blastocyst. Then, if you are asked after implantation what occurs, this is your answer. After implantation, the chorion develops the finger-like outgrowths called chorionic villi. Then the endometrium, together with the chorionic villi, forms the placenta. The umbilical artery and the vein develop inside a hollow tube to form the umbilical cord between the fetus and the placenta. If you see it's for more marks, like 10 marks, then you can actually say what the function of the artery and the vein is. But this is a new question now, so read carefully. One can say till fetal development, this is the answer, and if they say till implantation, that was the one we did in slide 21. Right, let's look at the nervous system. It's 54 marks. So know the divisions of the central nervous system and peripheral nervous system. The parasympathetic and sympathetic systems are antagonistic. The sympathetic system might increase the heart rate, while parasympathetic will decrease the heart rate. What's our common mistakes? Cerebrum controls voluntary actions and is responsible for interpreting sight and hearing, whereas the cerebellum coordinates voluntary actions and is for balance. When it comes to a reflex action, say a reflex action is a rapid, you must say a response to a stimulus. Reflex actions are performed, you should know, by the spinal cord. When it comes to the pathway of a reflex arc, it's receptor, sensory neuron, dorsal root ganglion, dorsal root, interneuron, motor neuron, ventral root, and effector. Remember grade 12's, receptors pick up stimuli and convert the stimulus into an impulse. Whichever sense organ you asked, Remember to say the sense organ will pick up the stimulus is converted into an impulse, and then this impulse is interpreted by the cerebrum. Always remember the order: stimulus, impulse, interpreted by cerebrum. The eye. Remember, ciliary muscles are used in accommodation. Circular muscles are found in the iris, and they're used for pupillary mechanism. Make sure you know this. For dim light, bright light, make sure you know these answers. And for distant vision and near vision, learn this well. Learn the labels, learn the answers well. Don't go into your exam on Friday without knowing this. The ear. We should know the oval window transmits vibrations to the inner ear. The round window absorbs excess pressure from the sound waves. Know the function of the eustachian tube as well. Remember, the eustachian tube maintains equal pressure on either side of the tympanic membrane. Now, for balance, you might be asked the cristae or the macula. Here are two set answers that you can actually memorize. So if they ask you something, you can just write this answer down. I've also included most of the exams hearing was coming out. So here's a set answer to remember if you asked hearing. And 332, I've taken the adaptations of the semicircular canals and I've put it here. And if you look how it's set out, how the structure helps the function. So it's surrounded by bone. Why? Bone is hard. It's there for protection. So make sure, if you don't know, they ask you a new adaptation, always write how the structure helps the function. Endocrine system. Know the positions of the glands. You must know the functions of the hormones. And make sure, when you're talking about the hormones, you're always using the word more and less. Make sure you know the negative feedback mechanism between TSH, thyroxine. Know ADH, aldosterone, insulin, and glucagon. Things to remember. Thyroxine is responsible for your basal metabolic rate. That's the rate of cellular respiration. Insulin converts excess glucose to glycogen, which is stored in the liver. And then glucagon, make sure you spell correctly, increases blood glucose level, and insulin decreases blood glucose level. Never say sugar. Homeostasis. We know homeostasis is to maintain a constant internal environment. Now, whatever situation they put you in or they ask you, hot day, cold day, make sure you're always using the words. If you look at this here, highlighted in yellow, more, 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 more. When it's a cold day, it'll be less, 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 less. Try your best to remember this. This is the easy place to lose marks. Then, regulation of carbon dioxide. I've included the steps here of what happens when your carbon dioxide levels in the blood increase above normal. The receptor cells in the carotid artery in the neck are stimulated. There are also receptor cells in the aorta as well, if you want to add it here. They send the impulses to the medulla oblongata. Medulla oblongata sends impulses to the breathing muscles, which is the intercostal muscles and the diaphragm and the heart, to increase the rate and depth of breathing, and the heart beats faster, and by doing that, more carbon dioxide is taken and exhaled from the lungs, and the carbon dioxide levels return to normal. And lastly, plant hormones. Make sure you know apical dominance. Know phototropism, know when lateral branches form. I've included an answer here. There's a set answer for B for phototropism, and this is when there's equal or uniform light. Then for geotropism, know what happens in the roots and know what happens in the stem. All you have to do is memorize this answer, and whatever happens on Friday, you can quickly write them down. Well, grade 12s, thank you for watching this video. I wish you all the best in the exams, and I hope and trust that this video will help you, and I wish you all the best in your future. Thank you.